In this video, I'm going to teach you how to perform proper aseptic techniques. First and foremost, you want to remove any clutter from your bench. You only want the items that you need to perform the lab exercise on your bench. Any personal items, you would want to stow them away. Then you want to Lysol your bench. Get a very good adequate coverage of the Lysol there. And then wipe that down with a paper towel. Now notice that I'm not wiping this dry. You actually need to leave the disinfectant on there long enough for it to work, so disinfection does take time. So let that air dry, don't wipe that dry. And the paper towel, that can go in the normal trash. That's the only thing that goes in the normal trash. Anything that is potentially contaminated or contaminated, including your gloves, will go within the biohazardous waste bin. All right, now that our bench is air dried, we now want to grab our materials that we need for the lab exercise. And if you're grabbing a glass tube, always bring your tube rack with you to bring the glass tubes back to your bench. And so we have our materials here. Next, we want to label. And before you do anything, you want to label, but you're never opening any of the materials. That's part of Aseptic Techniques, is to not open any materials until you actually need to open them. So with our label, we want to label first before we inoculate the medium, because you might get confused. Well, wait a second, which bacterium did I put on there? I can't remember. And you don't want to do that, right? So you always want to label first. Now we need 5 things on our label. You need the bacterium's initials, you need the incubation temperature, you need the medium's initials, and the 2 easiest things you can't forget, today's date and your initials as well. Now, how do I know the bacterium's initials? Well, I'm going to take the culture from which I'll be getting an inoculum, and this label here says that this is Escherichia coli, and I'm going to take a look at my lab manual and find that the initials for Escherichia coli are EC. So I would put EC on my label here. And then how would I know the incubation temperature? I would need to read through the entire lab procedure, and at the very end you'll find where we're going to, what temperature we're going to incubate these materials at. And how would I know the medium's initials? Well, there are different types of media, and so your initials will differ based off these types of media. This here is an agar. It's a semi-solid type of material, and this, for sake of example, just to show you what an agar is, this is not aseptic, right? I have everything open, I don't need to have it open. But to show you what the agar is, that is your agar right there. So with my agar, how I know the initials of the agar, there are different ingredients in different agars. And so my initials will always contain A, because A stands for agar. But how I know what type of agar this is I would look at the lines on the side of the petri dish, the plastic dish that the agar is within, and I would look at my lab manual, and I would find that 2 lines stands for TSA, or tryptic soy agar. Now if I had 1 line, that would mean nutrient agar, or NA. Now a broth, though, you would always have a B in the initials, because B stands for broth. And now notice that on the sterile broth, we don't label the glass, because that's a nightmare to get that cleaned off. So how would you know if this is nutrient broth or tryptic soy broth? You would know that based off of the label that's on the tube rack from where you're getting this sterile medium. Okay, now we want to label this plate. We want to go from an E. coli broth culture onto a sterile medium that is a tryptic soy agar. So now we know that from here we want E. coli, so I'm going to put that onto my label. And then the incubation temperature, I read through the lab manual, and I know that we're going to incubate at 30 degrees. I know that this is a tryptic soy agar, because I see those 2 lines there, so a TSA. I'm going to put today's date. And then I'm going to put my full initials. And the reason why I recommend your full initials is because sometimes students have the same initials, and so you don't want to confuse your materials with other students. All right. Now, also notice that because this is the first lab, it can be very confusing. And when students start labeling, sometimes they'll take the exact label from this culture, verbatim, and put that onto this sterile medium. There's a couple problems with that. The reason why is because this is a broth. This will have B in the initials. This is an agar. This will have A in the initials. So if this were labeled NB, but I'm putting it onto a TSA, I wouldn't want to label this NB, right? Also, let's assume that I incubated this at 37 now. but I want to incubate it at 30 degrees. So I don't want to put meat at 37 degrees today. That's not the condition at which I'm going to incubate it. So really the only thing that you're getting from the label, from what the culture, the inoculum from the culture that you're really just getting the bacterium's initials is all you're getting. And always be thinking when you're labeling your plates, what is it and where is it going? In what conditions am I growing this bacterium? Also notice that when you're labeling, that you're labeling the agar side and along the edge there as well. So the agar side, I've already showed you the agar here, but the agar side, but not the lid. And the lid gets shifted, and that matters in other types of labs. Doesn't matter today, but it matters in other types of labs. So because lids get shifted, you never want to label the lid, you always want to label the agar side there. All right. Now, if I were labeling a glass tube, then I don't want to write directly onto this glass tube. Remember, it's a nightmare to get all of that off. And so instead, I'm going to label a sticky label. Notice that when you're grabbing the glass tubes, that you always grab them by the glass and never the cap. And the reason why is that sometimes those caps are loose. So it's actually a very good practice to pick up the tube by the glass and just go ahead and tighten that cap just to be sure. If it's loose, it's not loose, it doesn't matter, just tighten it just to be sure. Also, never tilt a tube because you'll dump stuff all over the bench, dump culture all over the bench or even yourself, so it's a safety issue. So if I actually had taken a sticky label and I put it onto this glass tube, I now have to label it. So I'm going to tilt it on the bench, and you don't want to do that, right? So I actually want to label the sticky label first. Take that label off, and then put that somewhere in the middle. Not too far up on the neck of the tube and not too far down. Somewhere in the middle right there. And then you should be good to go with that. So now let's label a sterile broth. I want to take an inoculum from an agar, a culture that's on an agar, and inoculate a sterile broth. So I need to figure out which bacterium is forming this culture here, and it looks like it's E. coli. So I'm going to put E. coli on my label. I looked at the tube rack that this sterile broth came from, and it said NB, so I'm going to put NB on my label. I'm going to incubate at 30 degrees. I'm going to put today's date and my full initials. Okay, good. So now we have both our media labeled, and we're ready now to actually inoculate the media and form these cultures. And we're going to do this aseptically. Now, I've already mentioned that you don't want to have things open all the time if you don't have to have them open. And this is a big part of aseptic techniques, that you're always keeping things closed as best as possible. You're also staying very close to the heat source, this incinerator right here. So it's hard for the first lab because it can be cumbersome because you don't know what you're doing, what is going where, and I'll help you with that, that's okay. But it's also cumbersome because you're having to use both your hands. It requires a lot of dexterity. You have gloves on if you're not used to wearing gloves. And then also you have to use both your hands at the same time, but I also have to use this plate and this tube. It gets a little cumbersome to do that, but I'm going to show you how to work around that. So first I'm going to take my loop and hold it like a pencil. And then I'm going to put the metal of that loop up to this screw right here into the barrel of this incinerator, this micro incinerator. And notice that the loop is not scratching the inner part of the barrel as best as you can. Try not to scratch that. Also notice that I'm not sterilizing the screw that holds the loop onto the handle. So because that's not sterilized, you want to, as best as possible, try not to get that into the glass tubes. But I'm going to hold this loop in the micro incinerator for about 5 to 7 seconds. And now I need to let that cool a little bit. Because if I grabbed my culture, especially from a broth, I grabbed my culture, you'd hear a sizzle, and then you killed them. That's them screaming, so you would need to start all over again. So let this cool a little bit. But notice how I'm also staying pretty close to my heat source. All my materials are staying close to the heat source. And I haven't opened anything yet, right? So I want to go from a broth, let's start from our broth, and then I want to inoculate a sterile agar. You can also set that down by the micro incinerator close to the opening where the heat is coming out. The heat's radiating out of that to minimize any contaminants. So you can set that down very nicely. Whenever you're working from a broth, you always want to agitate the broth. to get a good representative sample of those cells. So you're going to twist your hand at the wrist right there. Never tilt it, right? That's the safety issue. Never tilt that to agitate that. So agitate it with your wrist. Okay, my loop is probably cool by now. In my dominant hand, I grab the loop and hold it like a pencil. In my non-dominant hand, I grab the glass of the tube. In my dominant hand that's holding the loop, I'm going to take my ring finger and pinky and grab the cap and hold the cap down. And I'm going to unscrew the glass. That matters in a second. Now notice that I have the cap held down in my ring finger and pinky. I don't put it down on the bench. I don't hold it up on the bench like that. That's not part of aseptic technique. So I always want to keep it in my ring finger and pinky. Also notice I don't recommend trying to hold it with your thumb and your index finger that's trying to hold your loop as well. That gets very awkward. So ring finger and pinky. Now every time I open a glass tube, you want to pass it very gently in front of the microincinerator. That radiant heat is going to help sterilize, try to keep any contaminants, minimize them getting into that broth there. So just very gently twist it back and forth, just a few seconds. All right, now I'm going to get my inoculum. I'm going to put my loop into that broth. And notice that I didn't put a screw in there, right? Because I didn't sterilize that screw. So as best as possible, try not to get that in there. I'm also trying not to flick the loop against the glass because that'll flick bacteria all over the bench or you. Before I close the tube, I want to, again, pass it in front of the microincinerator and then close the cap onto that tube. And notice that I'm twisting the glass and not the cap. And the reason why that matters is because that inoculum will fall off onto the bench or onto you. And again, it's a safety issue. So go ahead, close your tube by rotating the tube into the cap. So now my inoculum is on my loop and I want to inoculate this sterile agar. And I recommend starting agar up. We've labeled agar up. I recommend starting with the plate on the bench agar up. And so when you start agar up, you just need to grab the agar portion of the plate and lift it, leaving the lid down and rotating your hand to now expose the agar to inoculate. Notice though that I'm staying very close to my heat source because I've exposed the agar, I've also exposed the lid. And if I get contaminants on there, then this is worthless. This is not aseptic techniques, right? So I have to stay very close to this incinerator. Now with my inoculum, I'm just gonna lightly drag my loop down that agar. And it is a semi-solid surface, it's like a harder jello. You're gonna feel probably a little bit of a resistance, but notice that my angle is a little flat. If I have it really sharp, a sharp angle, I might actually rip into that agar. You don't wanna rip into the agar. So just lightly drag it down, that's all you need. Rotate your hand again, close that plate back up, and now I still have this loop that's contaminated, so I'm gonna go ahead and put this in the microincinerator for 5 to 7 seconds. And then that's it. You've actually inoculated a sterile agar plate from a broth. Now, let's actually inoculate a sterile broth from a culture that's found in an agar plate. Now, when we've already labeled this, now when you're grabbing an inoculum from an agar plate, students usually get very overambitious with this and they grab a lot of inoculum. And it's hard to wrap your head around that there are millions of cells on this plate and you don't need that many of them. And it makes it hard to thin them out and spread them out in future lab exercises if you grab a huge inoculum. So you really only need to just barely nick the growth of the bacteria. And you just need like a comma-sized amount. So let's sterilize this again just because I've been talking over it. So we're going to put that into the microincinerator, 5 to 7 seconds. I'm going to let that cool. Now I'm going to grab the loop in my dominant hand. With my non-dominant hand, I'm going to grab the plate, the agar side of the plate, rotate my hand, and just lightly touch just a little bit, just nick it just a little bit. Close the plate up. And notice now my hand is free to grab this sterile broth. That's why I don't have to have everything open all the time at the same time, right? Now my hand is free after I got my inoculum from that plate. Now I can grab the sterile glass tube, ring finger and pinky, rotate the glass tube, sterilize the mouth of the tube, put the inoculum into the broth. I'm going to shake that a little bit just to get the inoculum off the loop. You don't have to go crazy with it. Sterilize that, close the cap. sterilize my loop again. So notice that you're always sterilizing your loop before you get an inoculum, after you inoculate. Also notice that you're always sterilizing the mouth of the tube after you open the cap and before you close it. And these are part of your aseptic techniques. Sterilizing your materials, not opening them when you need to, or until you actually need to, and then staying very close to your heat source here. Now that we've inoculated our materials, we're ready to incubate them. Now for your plates, you're going to incubate auger up. And so a couple different reasons for this. You labeled auger up, you labeled the auger side. So it's easy to find your plates out of the incubator if they're labeled that way and you see it that way. Also, more importantly, you should incubate auger up, because if you incubate auger down and there's condensation on the lid, the condensation might get down onto the bacterial growth, and now it's a community swimming pool where it's hard for you to get them separated from each other if it's a bunch of liquid on that growth there. Also, if there's any contaminants on the lid and you incubate auger down, that contaminant can fall onto the auger and grow and contaminate your culture, and so then it's worthless at that point. So you're going to incubate auger up. If you have multiple plates, you can actually rubber band them together, and then you can stack them about 2 to 3 high in the incubator. So don't feel like you have to have a mono layer, one single layer of a bunch of plates of all the students in the class. You can actually stack them a little bit. And then when you incubate your broth tube, you're going to take your tube rack with your medium that you just inoculated and carry that over to the incubator, and there'll be a tube rack in the incubator, and you just set that in there, and then that's it.